I am Sandhya, Assistant Professor of Chemistry, Department of Chemistry, JJ College, Ordnance Science and Pudukkottai. Today, classify carbohydrate and structures and then classifications. The term carbohydrate is derived from the first as the French is a hydrated carbon. Compounds composed of a CHO, then carbon, hydrogen and oxygen. CH2O and when N is equal to 5 then C5H10O5. Not all carbohydrates have this empirical formula. Deoxysugars and amino sugars. Carbohydrates are the most abundant compounds found in nature. Then cellulose 100 billion tons as a joule. Carbohydrates is a polyhydroxyaldehyde or ketones or substance that yield these compounds on hydrolysis. The group glyceraldehyde dihydroxyacetones then keto group both can be written as C3H6O3 or CH2O3. Carbohydrates with the aldehyde group of aldose, carbohydrates with the keto group of ketose. Empirical formula for many simple carbohydrates is CH2O and hence this name hydrate of carbons. The characteristics of the carbohydrates, most carbohydrates are found naturally in bound form rather than as simple sugars. Polysaccharides, starch, cellulose, and gums. The cycloproteins and proteoglycans, hormones, blood group substances, antibiotics. Glycolipids in the cerebrosides, glycosides, and mucopolysaccharides, and then the hyaluronic acid, then the nucleic acid is functions of these reactions for the storage form energy in the body and other than cell surface recognition respirator by other cells, hormones and viruses. The next, cell surface antigens. For example, the blood group, garlic, glycoprotein, mucin contains more than 60% of carbohydrates. The glyceric glycoproteins machine contain more than 60% of these carbohydrates than these proteins. Yeah, structural component of many organisms. The first one is the cell walls of bacteria, exoskeletons of insect, another than the cellulose of plants. Next point is of the intermediates in the biosynthesis of other basic biochemical intensities in fat and proteins. Other than associated with other enzymes such as glycosides, vitamins, and antibiotics, precipitates in biological transference, cell to cell recognition, activations of growth factors, modulations of the immune system, lubrications of skeletal joints. Other than the classification and then nomenclatures of the carbohydrates. First one is the monosaccharides. Monoses are glycoses. Example for trioses, trisoses, pentoses, hexoses. Another than oligosaccharides, di, tri, tetra, penta, up to 9 or 10. Now, most important are the disaccharides, polysaccharides or glycans, homo and heteropolysaccharides. Then complex carbohydrates. Another than derived carbohydrates, oxidation products as ascorbic acid, reducing products like glycerol, amino sugar is glycosan, deoxy sugars and deoxyribose. And then monosaccharides, polys, polyhydroxyaldehyde or ketone that cannot easily be further hydrolyzed. The simple sugar, number of carbons are named for this 2, 4, 5, 6, 7, 9, triose, tetrasos, pentasos, hexasos, nanosos. Mm, The triose is a glyceraldehyde, tetrose is a erythrose, pentose is a ribose, hexose is a glucose and fructose. For example, heptose is pseudoheptalysis and anose is a neuronic acid. The classifications according to the functions group is aldoses and then ketoses. Function group sugar class number of carbons and number of sugar aldehyde and then ketones aldehyde then the aldose 3 4 5 6 glucose erythrose ribose glucose galactose mannose then ketose 3 4 5 6 and then 7 dihydroxyacetone erythrolose cellulose fructose pseudoheptulose. Example for this monosaccharide glucose, fructose, graph sugar. Glucose is the most important carbohydrate in the biologically. The cell uses it as a source of energy and a metabolic intermediate. Glucose is produced in the process of photosynthesis and is used in both prokaryotes and eukaryotes. Fructose is a levels of fruit sugar found in honey and fruits. Fructose is produced from the digested in a sugar. A disaccharides are consisting of glucose and fructose. Fructose is the sweetest naturally occurring sugar, estimated as twice as sweet as sucrose. This then the galactose, it is a sugar found in the dairy product in the form of lactose. It is a disaccharide formed of glucose and galactose. It forms part of glycolipids and glycoproteins and several tissues of the body. Then other than the oligosaccharides are hydroxyl polymers of 2 to 6 monosaccharides composed of two monosaccharides, for example sucrose and lactose. And the disaccharide is produced from the condensation of two monosaccharide molecules. The lactose, milk sugar, is formed in the mammary gland. It's composed of galactose and glucose. Maltose is a malt sugar is composed of two molecules of glucose. Sucrose is a table sugar, cane sugar, beet sugar is composed of glucose and then fructose. Disaccharide sucrose, the most widely occurring disaccharides of ordinarily table sugar. Structures of the glycosidic linkage, beta glycosidic linkage. Then maltose structures, then the solobias structures, the lactose structures. Lactose is reducing sugar, the hydrolysis, they yield the D-glucose and the D-galactose is the glycosidic linkages beta. The oligosaccharides, it occurs in glycoproteins, which are proteins to which oligosaccharides are conveniently attached. Trisaccharide, tetrasaccharides, pentasaccharides. A trisaccharides, it will be the ribose, fructose plus galactose plus glucose. Tetrasaccharides is saccharose, 2 galactose from then glucose and fructose. Pentosaccharides is verbenose, 3 galactose and glucose and fructose. Polysaccharides are homopolysaccharides, heteropolysaccharides. Then first one homopolysaccharide, polysaccharides that are polymers of a single monosaccharide. Those made up of more than one type of monosaccharide than the heteropolysaccharides. Glucan, yeah, homopolysaccharide consists in glucose monomeric units. Galactan, yeah, homopolysaccharides consisting of galactose units. Three important polysaccharides are all of which are glucans, glycogen, starch, and then cellulose. Then starch, heating starch with water produces amylose. 10 to 20 percentage of amylopectin, 80 to 90 percentage. In amylopectins, the chains are branched. Branching take place between the C26 carbon 1 at intervals 20 to 25 glucose units. Then amylose. The amylose chain form a calyx forms. This case the blue color change on these reactions with the iodine. Amylose is poorly soluble in water but forms as Malaysia suspensions. The partial structure of amylopectin. The molecular weight of about 1 to 6 million include hundreds of interconnecting chains, 20 to 25 glucose units. The amylopectin is cases a red violet a change on this reaction of this reaction. The change is usually masked by reactions much amylose to iodines, amylopectins unit. Glycogens. The glycogens chains are much more highly branched. The molecular weight is very high. Polymers of alpha 1N4 linked subunits and branched is alpha 1N6 similar to the amylopectin. Highly branched. The size of the structure of glycogens units as its functions. First one is the size makes it too large to cross cell membranes. The structure of a glycogen solves the anomalous of osmotic pressure within the cell. Third one is the high branch structure of glycogen simplify the cell liturgical problems. The glucose from glycogen is a heavy water soluble in an ideal source of ready energies. Cellulose Yeah, proton of cellulose structures the glycoside linkage 1 to 4. The special properties of the cellulose, then the outside OH group is ideally situated digit the chain made together by forming the hydrogen bond. Zipping many cellulose chains together is a way gives a highly insoluble cell. Cellulose is found in the first from the seeds hair of cottons. Wood cellulose, on the other hand, forms on the composites with linen and then polysaccharides. The bacterial cellulose, glyco Glycanobacteria are involved in the production of a very peculiar form of cellulose. Cellulose acetate is ester of cellulose. Ethyl cellulose is a microcapsulated drug delivery systems. Hydro Hydropropyl cellulose is soluble both in water and organic solvent, used as a lubricant, corneal erosions, neuropathylytic keratins. A textran in bacteria and yeast made alpha N 6-linked polyglucose and heavy alpha 1N3 branches. Synthetic textrans are used in several chemical producers, for example, substrates. Cetin linear homopolysaccharides of N-acetyl glycosides reduced in beta in 1,4 linkage, and then heteropolysaccharides in the body of ants glycomygens. And these molecules are long unbranched polysaccharides containing a repeating disaccharides unit. GAG are highly negatively charged molecules with a extended formation. They impart a high viscosity to the solution. Gates are located primarily on the surface of cell or in the extracellular matrix, then ECM. Along with the high viscosity of the GACs comes low compressibility, which makes these molecules ideal for the lubricating fuel in the joint. Other than at the same time, the rigidity provides structural integrity of the cell and provides a passing base between cells, allowing to cell migrations. The disaccharides units contain the either of two modified sugar called amino acids N-acetyl galactosides, N-acetyl glucosides. The acetic sugar uric acid such as glucuronic acid and iodoronic acid. The amino group is usually acetylated. This eliminates the positive charges. Some groups also very negative charged, very high density of this reaction. So keratin sulfate is an expectation with galactose expression instead of an acetic sugar, as it does not contain the sulfate by hyaluronic acid. For structures for these reactions, the linkage for this protein core, the classification of glycoamylins. First one is monomeric, second locations, a type of glycosidic chain length of the disaccharide, nature of the core proteins, the tissue distribution is their biological functions for this reactions. This case physiological significance for specific reactions for this reactions. Lauric acid or structures acid structures is however the compounds hyaluronic acid is a non-velocity formed complex with the protokinase and magnesium. So only GAG presents both in animals and then bacteria. The specific function acts as the lubricants and the shock absorbers. Jontorant sulfate is made up of glucuronic acid, N-acetyl galactosine substrate. Most abundant GAC is cartilage, tendons, ligaments, heart values. A heparin is a composite alternating unity of N-sulfonic, N-sulfo D glucose and 6-sulfate and glucuronated phosphate. It is an intercellular stage of this reactions. Specific function. Specific function is a heparin and warfarin are widely used in the treatment of thrombotic and thermometabolic conditions such as deep vein thermobiasis and pulmonary emboliosis. The dermatan sulfate is contribute the skin, blood vessels, and then bear. Keratin sulfate is contains D-galactosamine N-acetyl galactose 6 sulfate is hernia, bone, for these reactions. In various types of arthritis, protoglycans may act as the adaptogens, contributing to the pathologic features of this condition. The amount of jontrotin sulfate in catalytic diminishes with the age. The other polysaccharides and then sitting structure analysis of these polysaccharides are for information on polysaccharide structures, the monosaccharide component, sugar linkage type, sugar sequence, monosaccharide configurations, alpha and then beta, D or L, molecular weight, amount and position substituents in the degree of branching. The monosaccharide component, the high pressure performance liquid chromatography, then sugar linkage type GCMS gas chromatography, and the physical methods of sugar linkage type monosaccharide configurations are substituents use degree of branching for this enema. So nucleomagnetic resonance. The physical methods for FTIR machines monosaccharide configuration substituents units for transformation infrared spectroscopy. The physical method is MS mass spectroscopy, sugar linkage type monosaccharide configuration substituents units degree of branching molecular weights. So sugar acids, deoxy sugars, monosaccharides in which an OH group has been replaced by HS group. Glucose salamines is amino sugars, is a fibers found in food delivered from plants, include polysaccharides such as cellulose, hemicellulose, pectins, gums, and lignans. Also includes non-polysaccharides such as the lignin, proteins, and tannins. Fibers are not a source of energy in non-ruminants because digestive enzymes cannot break down fibers, but ruminants can too. The bacteria is a non-ruminants GI tract can break down some fibers. The classification of fibers is soluble by the decay glucose and then so lower blood glyceraldehyde. Second one is insoluble fibers. The action in this body is acrylates, storage, hydrolysis. Is requirements for carbohydrates. Carbohydrates are essential nutrients are not then essential nutrients because the carbon skeletons of amino acid can be converted into glucose. However, the absence of a dietary carbohydrates leads to ketone body production and degradation of body protein constituent amino acid provide carbon skeletons and glycogens. The glycemic index for this reaction, the ability of the body to digest different carbohydrate can be described by the glycemic index. So, low GI foods release glucose more slowly and steadily. High foods cause more rapid rise in the blood glucose level. So GI means glycemic index. So thank you.